cells, the father's X chromosome becomes inactive. In the other 250 cells, the mother's X chromosome becomes inactive. Okay. So one of the two X chromosomes becomes inactive. Once it's inactivated,
so then when this female has progeny, does she automatically transfer the open X chromosome, or can she?
It gets reactivated
during
the production of gametes. So she resets the stage every time during meiosis.
Cool.
So this happens at random. Okay. So let's look here at random. Okay. Okay, so this happens at random. So let's understand first the mechanism, the molecular mechanism of this process. And once we understand the molecular mechanism, you'll see the consequences of it with regards to the genes that are on the X chromosome. So the mechanism by which this happens is that there is an RNA that is made. A specific RNA is made called XIST, X-I-S-T. This is made from the X chromosome. It coats the X chromosome. Literally, small RNA molecule, well, it's not really small, it's quite large. So there are a few kilobases long. This RNA is made from a gene on the X chromosome or from a locus on the X chromosome, and it actually coats the entire X chromosome, the RNA. It binds to the DNA and the chromatin on the X chromosome, and this leads to the inactivation of the X chromosome. So here is the X chromosome, here is the gene XIST. It's active in this chromosome. It makes an RNA, and that coats the entire chromosome from which it was made. It works in what we call cis. Basically, the RNA is made from a gene on this X chromosome. It will only coat the chromosome from which it was made. Turning on this gene happens at random, at that stage, 500 cell stage. So at the 500 cell stage, there are two chromosomes, correct? The promoter of this XIST gene becomes active in one of those chromosomes at random. The RNA is made. The RNA literally spreads like an inchworm across the entire chromosome, coats the entire chromosome. Other proteins now bind, and you get this bar body form, and you become inactive. One of the consequences of this binding of the RNA is the DNA becomes inactivated.
In autiploid individuals like XXY and XXX,
you come to that. Very good question. You have multiple bar bodies. Exactly. Now, what happens on the X chromosome that's going to stay active? Well, one possibility is that this is the way you inactivate it. Whatever is not inactivated is active. That's one possibility. But it's never as simple as that. On the active X, you have the same gene. XIST and E6 are the same gene. In XIST, you're making RNA from left to right. In E6, you're making RNA from right to left. It's the same gene. It has two promoters on either side of the gene. In one instance, you make RNA left to right. In another instance, you make RNA right to left. An RNA. Now, in this X chromosome, where the chromosome is going to remain active, you also make RNA from left to right. And these two RNAs pair become double-stranded RNA. And because they become double-stranded RNA, you inactivate this RNA. It doesn't spread and coat this X chromosome. And so this X chromosome stays active, this X chromosome. That's how you see this active state and inactivate this X chromosome. In the whole process, once the XIST RNA binds, you methylate the DNA. You methylate the DNA. Once the DNA is methylated, that chromosome stays inactive for that. And the reason is, once you methylate DNA, it is copied during replication. There is a mechanism by which you can copy. You put a methyl group And usually the metal group is put on cytosine in DNA. So you put a metal group. Now you go through replication, you have one DNA strand with a metal group, the other DNA strand without a metal group. There is a specific enzyme called DNA methyltransferase that looks for this. When it finds a molecule that looks like this, it will methylate the other C and become C. So it replicates itself. So once the state is set up, you can replicate the state and inherit the state over and over and over again. And that's how you maintain the state once it's established. Metal groups, you guys don't have to know the structure of it, but I'm just showing it to you in case you're doing biochemistry next. You guys should know what a metal group looks like. So the methylation status is maintained during replication and mitosis, and that's how we inherit the state. Okay, so what are the consequences of X inactivation with regards to genetics? That's the question we are interested in. So, here's what's going on, right? You have an egg and a sperm. They get fertilized to form an embryo. The embryo divides until it reaches a certain stage. And it's at this stage here that you're going to make a decision to, at random, inactivate one X chromosome or the other X chromosome. It's a purely random process, probabilistic process at this stage, right here. So, here is the female, the egg, sperm, the zygote. So, I'm showing you the female's X chromosome as a black chromosome, and the male X chromosome has a gray chromosome. We're going to produce an embryo. Right? The cell divides many, many times, and you're going to produce a multiple cells, 250, 500, 750,000 cells. At this point, at random, in each of the cells, you're going to inactivate one of these X chromosomes. And it's a random process. About half will go one way, half will go the other. And so in this cell, for example, it's the father's X chromosome that gets converted into a barn body. In this cell here, it's the mother's X chromosome that gets converted into a barn body. Similarly here, and here again, the father's X. So you have this random inactivation taking place of the entire chromosome, almost the entire chromosome. And so the embryo really is a mosaic. It's got patches of cells where one X chromosome is active and the other is inactive. But then there are patches of cells where the other is inactive and the other inactive. Right? So this is the important thing is the genes are all the same in all of the cells. No difference. But because of this inactivation of the chromosome, the expression of the genes varies. It's a developmental rule that sits on top of a Mendelian rule. And that's why we call it epigenetics. It's on top of genetics. But the question?
When the individual is chromosomy with 3 X's, the two are put into heterochromatin X will expressing enough genes to affect the individual?
Yes. So notice I always keep saying almost all of the genes on the X chromosome are inactivated. There are a very small number of genes called escape loci that escape from the mutation. So if you have multiple X chromosomes, not the usual 2, 3, or 4, you have certain syndromes, correct? Klinefelter, etc. The reason is those additional genes that are escaping the X chromosome, they're being made more amounts than they normally would. That leads to certain syndromes. Okay, so Bauer bodies. When you have 3 X chromosomes, 2 out of the 3 X chromosomes will get packaged into a Bauer body. That was the question that was asked. If you have females with 4 X chromosomes, 3 of them will become Bauer. If you have a male with XXY, then you will have one Bauer body in the male itself. If you have an X or female, of course, there are no Bauer bodies. And notice there are syndromes here. You can say, well, you're inactivating the entire X. Why should that give you a syndrome? The reason is it's not the entire X that's being inactivated. There are small segments of the X chromosome that's inactivated that are actually active, and that's important. And those are all escape loci. So, X female, 1 bar body. XX females, 2 bar bodies. XY male, no bar bodies. XXY male, 1 bar. So the cell has a mechanism that always basically compensates X chromosome number equally restricted. So now what happens when you have bar body formation with regards to the expression of particular genes that are present on the X chromosome? So you have a male X and a female X, correct? You're going to divide male X, female X. And then, at random, you're going to inactivate one of the X chromosomes. In this instance, you're inactivating the male X. In this instance, you're inactivating the female X. And then once that decision is made, you inherit the same inactive X chromosome. So you inactivated the maternal X. Now the cell will divide further. You still get 2 X chromosomes. This cell will divide further and give you 2 X inactive, but those are the fathers that are inactive. And so you're constantly inactivating one gene out of the 2. So what happens? You've seen tortoise shell cats. All of you know about tortoise shell cats. The gene for the coat color of a cat is on the X chromosome. Okay. So, and the pathway is very simple. You go from black to orange. And there is an enzyme called the enzyme O that converts black to orange. So, the O gene is present on the X chromosome. Female cats that are heterozygous for the O gene are tortoise shell. And it's only the heterozygous females that are tortoise shell. According to Mendel's rules, these cats should either be orange or black, depending on what is recessive and what is dominant. But that's not what you see. The cats are neither, they're tortoise shell. So how is that happening? Why is that happening? So let's look at the X chromosome and how it behaves. So you take an XY male. If it's normal, the O gene is present on the X chromosome and the male is orange or ginger. If it's mutant, the male is black. Kind of makes sense, correct? Because you go from black to orange. If the enzyme is being made, you're going to be ginger. If the enzyme is not being made, you're going to be black. In females, if you're homozygous OO, you're ginger, because you're making the enzyme that converts the black pigment to the orange pigment. If you're little O, you're mutant, you're black, because the genetic pathway breaks in half, and you stay accumulating the O pigment. But why are the heterozygous females tortoise shell? Right? That's the question. How does that happen? It's because of X inactivation. So let's do a cross. Taking an OO female cross to an OY male. Right there. You're getting a female F1 that's heterozygous. Big O, little o. So now let's remember the pathway. This egg that's been fertilized to produce this heterozygous female is going to divide. The cells are going to divide. Okay? So here are your 2 X chromosomes. Right? One is mutant, one is wild type for the O gene on the X chromosome. One of these X chromosomes is inactivated. In this example, this chromosome is inactivated, and you get a ginger patch of a cell that's going to be active, because this is the active gene, this is the inactive gene, and you're going to be ginger colored. In this cell, during that stage when you make that decision to form a bar body, this gene is going to be turned off, this gene is going to stay on, you're going to be a black cell. Now these cells will divide further. You get 4 cells. But once the state is set up, it's maintained, it's inherited. And so now you have a patch of cells that are ginger, a patch of cells that are black. And then they of course divide further, and there are a patch of ginger cells and a patch of black cells. And that's what you see in your cat. You get a tortoise shell. It's not incomplete dominance. This is a completely different rule altogether. That's why I kind of keep incomplete dominance very far away from epigenetics and bar bodies. so this is basically how you get tortoiseshell cat. The same thing happens with color blindness. Green and red color blindness is a gene on the X chromosome. Females that are heterozygous can visualize color because in the cornea of the eye, there are some cells where the gene is active and some cells where the gene is inactive. So they are able to visualize color. They may not be able to visualize color to the same extent as somebody who's totally normal, but they're still able to visualize color because it's kind of. And this methylation pattern is inherited, though it's not inherited perfectly. So there were studies done on twins, identical twins. And you can see that here is a child's X chromosome with methylation. You're studying methylation patterns along the chromosome. And you can see they're very similar. But as they develop, the methylation patterns begin to change between one individual and the second individual, even though they're twins. And the reason is methylation is not inherited perfectly. And the environment does affect the inheritance of methylation. There's an example where the environment will affect your phenotype. And so because of that, methylation patterns of identical twins will show differences. So remember I told you, if you want to commit a murder, make sure you have an identical twin because your DNAs are identical. Well, honestly, we have ways to catch you even then. You can study your methylation. So don't do it. Okay, so that's one pattern that we talked about, epigenetic pattern, that affects the expression of genes that results in an interesting, totally unique phenotype that you didn't observe before. Now we're going to look at another pattern. So we were talking about genes on the X chromosome in this example, correct? Now we're going to look at a very small number of genes, about 200 genes, present on your autosomes, okay, on the 22 chromosomes, that exhibit something similar. Keep in mind, these are only a very small subset, 200 odd genes. They are present on your autosomes. They show something very similar. So, sperm, egg, embryo, divide, multiple cells, and what happens for these few autosomal genes is that a gene that comes from the father stays active, the gene that comes from the mother stays silent. So here, unlike Mendelian genetics, it determines which gene you got from your parents. It determines what's going to happen. Now, out of the 200 genes, a subset of genes follow this rule, where the active gene comes from the father and stays active in the child. The inactive gene comes from the mother and stays silent in the child. For the other set of genes, it's the exact opposite. The genes from the mother is active, and the gene from the father is inactive, and it stays that way in the child. And this phenomenon is called gene imprinting. You've imprinted a particular gene state, and it is maintained. Question? This is not the X chromosome. These are genes on the autosomes. It's completely different. Okay? It's a completely different example of that change, kind of change track. Finished with the X chromosome inactivation. We're now talking about genes on the autosome, and only a very small subset of genes. Only 200 genes in your entire 25,000 genes in your genome. Only those 200 genes exhibit this phenomenon. Okay. Any other questions? Okay, so this is known as imprinting. It occurs on the autosomes, and it occurs only on some of the genes on the autosomes. It's a very small subset of genes. So here is a famous example. These were sheep, isolated. Anybody know what Callipyge means? It's a Greek word. Anybody have an idea what Callipyge stands for? You don't understand, remember Greek? Anybody study Greek? No? Okay, it means beautiful bottoms. So, you see this sheep here, it's got a nice big butt. This is Callipyge. discovered in the 1980s by a farmer in Oklahoma. Farmers are like, yay. It's not good out there. Okay, so the farmer observed these sheep with big butts. And it's called beautiful behinds or beautiful butts. And so he basically makes money because sheep with large butts means lots of money, you know, leg of land. Right? So he's trying to think about it. Of course, scientists want to figure out why the hell are these sheep having big butts, right? What's going on here? So that's where we're going to try and start. We're going to see how this gene behaves, how this mutant gene behaves. Okay, so let's forget about all this. Let's make a cross. Let's do a cross. Classic cross, right? We're going to start with a male that's got big bottom. We're going to cross it to a normal, true breeding female. And you get a result. 200 big bottom males, 200 normal, 200 sheep that are big bottom, 200 sheep that are normal. So, what's the gene? Now remember, this is a true breeding female. So, and this is a gene on the autosome. I'm going to tell you this right now. So, what's the genotype of the male? Homozygous or heterozygous? Heterozygous, correct? Because they're getting a 1-to-1 ratio in a test cross. That's what they set up. Okay, so you say the male is heterozygous, the female is homozygous. And it's likely that the male, the mutant, is a dominant gene, correct? Because you're getting C over N and you're being calibrated, right? So it's most likely an autosomal dominant gene. That's what you would think from these numbers. 50% C over N, 50% N over N. And I told you already that calibrated is not on the X chromosome. It's sex independent. It's an autosomal gene. So you could easily, from this one cross, you could say it's an autosomal dominant gene. Now remember what Mendel did in his very first experiment? He'd taken a male yellow plant, crossed it to a female, whatever white color. What was the next experiment he did? A reciprocal cross. So scientists being good geneticists followed Mendel and they did a reciprocal cross. Now, they basically took a big bottom, crossed it to a normal male. Normal male, big bottom female. Now what would you expect in this cross if Mendel is right? You should get 50% calibrated, 50% normal. What you observe is 100% normal. None of the sheep had calibrated. Right? Surprising, right? Completely unexpected. What's going on here? Right? You could say, hey, calibrated is now acting like a recessive trait. That doesn't make sense. When it came from the male, it acted as a dominant trait. When it came from the female, it acts as a recessive. So what's going on? So they did a better experiment. They took a homozygous male, CC male, crossed it to a homozygous NN female. 100% calibrated, as you would expect. They do the reciprocal cross, homozygous NN crossed to homozygous CC, 0% calibrated, 100%. So it depends, the mutant gene, if it's coming from the father, the progeny can have it. If it comes from the mother, the progeny don't express it. So it depends on your parents. What they were determines what you are going to do. In this example, it is determined by the father. For other genes, other phenotypes, it is determined by the mother. So this is known as imprinting. Sorry, question? Imprinting. So the mutant gene is expressed in offspring when it is inherited from the male. The mutant gene from the mother is always silenced. The silence, basically. So if you do a cross of a normal female with a normal male, normal phenotype, the female allele is turned off. Let's imagine this red allele is the female allele. This allele is repressed. This green gene is made into RNA, and it is expressed. This is imprint. This is exactly the same thing. There's DNA methylation that's going to play a role in this process. In a different way, the DNA methylation is going to play a role in this process. So this gene is turned off in the female, it is on in the male, and that's why it's carried on in the body. That's the normal version, right? No mutation, but it's that gene. Now if you have a mutant version of this gene, you have a mutant version of this gene, you have a normal female crossed to a mutant male, this gene has a mutation in it, this gene is going to be turned off in the female, correct? So when this gene is turned off, this gene is on, and so all of the progeny will basically in the mutant version of the calibrated gene is going to be made and is expressed. On the other hand, if you have a mutant female and a normal male, this mutant calibrated gene is going to be silent. It's not going to be expressed. And because this mutant gene is not going to be expressed, only the normal gene is going to be expressed. You get a normal. So how does this mechanism work? So there are a small number of genes, about 200, present on the autosomes, where the allele from one parent is shut off and the other parent's allele is maintained in the offspring. So, and this decision is determined during the production of the egg and the sperm. So here you're making a decision during gametogenesis, during the production of meiosis, actually a step before meiosis, is where you decide, okay, I'm a male, I'm going to turn off all of the previous genes. If you're a female, you're going to keep it on, or vice versa. So, during gamete formation, you determine which gene is going to be on in which parent or not. So, in the male, the gene is maintained in an on state. In the female, the gene is turned off. Now, once that happens, of course, you form an embryo, the state is maintained. Then it's carried on through mitosis in the same way, and so you get a phenotype at the end. The original imprint is erased during gamete formation, and a new imprint is established during gamete formation. So it's during gametogenesis that the imprints were erased and the new imprint is set up, depending on whether you're male or female. And this is basically an example. And there are about 200 different loci. And they cause all sorts of interesting phenotypes as well. There are certain syndromes that happen because of mutations in imprint. So, I'm going to give you a different example, not the calibrated gene, but a different gene. This is a gene called the insulin growth factor. It's important in growth of individuals. And here is the gene. It's present from the father's chromosome and the mother's chromosome. There is an enhancer. It binds a specific transcription factor that will turn on this gene. Right? Just as we talked about, transcription factors binding enhancers activating the gene. This enhancer will bind this protein and activate the gene. Now, I haven't talked about something called an insulator. This is a sequence element that is usually located between an enhancer and a promoter of a gene. And that regulates the enhancer. In this one example, I'm going to talk about insulators. You guys don't have to remember anything about it. But it's important in biology. There is a protein that binds this insulator. But in the father, this sequence here is methylated. The DNA of this sequence on this sequence here is methylated, and this protein cannot bind because it's methylated. This CTCF protein is unable to bind, and therefore it's unable to block the enhancer from activating the protein. In the mother, this gene is inactive. And the reason it's inactive is this insulator protein is not methylated. The sequence here is not methylated. DNA is not methylated. this CTCF protein can bind. When this CTCF protein